Hi, I'm Witkowski here at Cold Spring Harbor Laboratory, and this is the 83rd Cold Spring Harbor Symposium on Quantitative Biology, the topic this year being brain and behaviour. I'm delighted to have an old friend of mine from England, from Cambridge, Michel Goedert from the LMB. Michel, we first met, we were thinking something like 1990?
Even before, I think, 1988 probably, that's the first time I met you, the Banbury Centre. There was a meeting on Alzheimer's disease, which was my first ever meeting on Alzheimer's disease, where we showed the presence of tau protein in the paired helical filaments of Alzheimer's disease. And the papers at the time had been accepted for publication, but they hadn't been published yet. So some of the work came as a surprise, I guess, to many of the people who were there, because they didn't know we were doing this. And today, 1st of June, it's actually 30 years to the day that the first of these 3 papers was published.
And so that was your first venture into structural studies in Alzheimer's?
Yes, it was. So Alzheimer's disease is defined by 2 types of abnormalities, so-called plaques and tangles. And it wasn't really known until the 1980s what they were made of. And so the plaques are made of this peptide called beta amyloid, and the tangles are made of tau protein. And this work we did at the time was the first direct demonstration of the presence of tau in inclusions.
And it was a very controversial field for quite a while.
Yes, it was. I mean, the first controversy was what the components were. Some people thought beta amyloid was also the major component of the tangles. And then after that was sort of clearer, the whole business of what was more important than what became an issue. And for that you need human genetics. And so in 1991 mutations in the amyloid precursor protein gene were described by other people. And only in 1998 tau mutations were found in familial forms of frontotemporal dementia. And so during these 7 years, working on tau was not the easiest proposition in the world. Now I think it's largely, well, the whole thing is sort of calmer, and I think as therapeutic targets, one believes that both are good.
So I seem to remember that one of the controversies was about how it was that nerve cells died. Did they die because of these proteins, or these plaques and tangles a consequence of something else, the cell dying?
I think that's in general for the field of neurodegenerative diseases as a whole, where you have abnormal protein deposits. The fact that the familial forms of disease, which in general are relatively rare, but you can find out the causes, that the causes are the formation of the inclusions, must mean that the formation of the inclusions, I would argue in all cases of disease, of neurodegenerative disease with such inclusions, that their formation is a bad thing. And probably the presence of these inclusions in nerve cells over long periods of time is sufficient to kill the cells.
Okay.
But, you know, it's unless you've found all the mechanisms that do this, it's always possible to say that it may not be like that. But I think the formal argument is quite strong. So you have mutations, as I'd already said, in the amyloid precursor protein gene, mutations in the tau gene, you have mutations in the alpha-synuclein gene that cause familial forms of Parkinson's disease.
Right.
You have mutations in the prion protein gene that cause Gerstmann-Streusler-Scheinker syndrome and some cases of Creutzfeldt-Jakob disease, and so on. So I think that's, I would argue that the ordered assembly of these proteins in the end causes these diseases.
Yes, right. So the fact you've got mutations in these genes producing an abnormal protein in the first place is indicative that that's the causative agent.
I think that's very strong evidence. I mean, what one then does, of course, because in general these cases are about less than 10% of the total, with the exception,
familial ones.
With the exception of Huntington's disease, for instance, which is always familial, but also has inclusions. What one does in the other cases, where it's like 10 or less %, one extrapolates to the sporadic forms, and the question is whether that is allowed. I would have thought that it is allowed, but then it's the question whether it's allowed to 100%, and that I don't know. But I think overall the evidence is quite strong.
So in 1988, were you doing electron microscopy? Were you doing structural studies wearing Klug of?
Yes, so the idea at the time was to, so it was a mixture. It was a mixture of biochemistry, molecular biology, and structural work. So the question was to obtain peptide fragments that one could relate back to the paired helical filaments one could only see by electron microscopy, but as soon as you take them apart, you can't see them anymore. So you needed a label that allowed to see them by electron microscopy and also by Western blotting. And that's where monoclonal antibodies came in. So Cesar Milstein was involved, and we're working in the lab where these technologies were invented. And so that's basically what the work did. It was the first direct demonstration. It was absolutely clear that these peptide fragments we got had come from the paired helical filaments, and so John Walker was there. He was doing protein sequencing on this material, and then we cloned so we got partial amino acid sequences, and we cloned the cDNAs and found it was tau.
So that work is something of a contrast with your current research.
Yes. So well, we then we went we were part of the work showing that the formation of these inclusions was relevant because of the mutations in the tau gene. Then we were part of the work showing the sort of prion-like properties of aggregated tau, which is still sort of very active field. And more recently, we used cryo-electron microscopy to obtain the high-resolution structures of the paired helical filaments from Alzheimer's disease. So that was published last year. And we've done more recent work on Pick's disease, which is not published yet, the same vein. And I think this work shows that there are different molecular conformers of aggregated tau in the different diseases, which some people also refer to as protein strains, and which one believes may give rise to the different diseases. Because there are lots of diseases where you have tau inclusions in the brain, and they're different diseases. Sometimes people ask, why are they not all the same disease? Because you have tau inclusions. And maybe that has to do with the fact that different molecular conformers that give rise to different diseases.
And is that similar to prion strains? That
yes, I think it's sort of formally, it's formally analogous, I would say, to something like that. And personally, I believe that ultimately, if you want to define a molecular conformer or a strain of a particular type, you need structural information at a high resolution level. So before the cryo-electron microscopy, it was only possible to get resolutions of like 20 or 30 angstroms for these filaments, and now you can get resolutions of like between 3 and 4. So I think that's a big step forward.
What things did the cryo-EM show you that were unexpected, or even that you had expected, and that confirmed your suspicions about?
One thing, I mean, for the Alzheimer field, I mean, one always, so tau has these repeat motifs in it, and one always knew that the core of the filament is somewhere in the repeats. What one didn't know, what part of the repeats. And so one surprise was that it's very clear that the core of the tau filaments in Alzheimer's disease is repeat 3 and 4 plus 10 amino acids after repeat 4. That the exclusion, for instance, of repeat 2 in that was unexpected. Most people would have expected that to be present, but it's not present. And in Pick's disease, there's only 3 repeat tau in the inclusions. That's what we see, so that was not surprising. But what is clear from that is what we see exactly what's present, and we can also explain why there are only 3 repeat tau inclusions, not 4 repeat, because the structure tells you that the extra repeat could not fit into that sort of structure. So I think it tells you quite a lot. It doesn't, it obviously doesn't immediately lead to any sort of treatments or so, but it gives you sort of a solid baseline from which to study, which is very rigorous and something you couldn't do before.
And what are you going to be talking about this evening?
So I'm going to talk about basically that, using cryo-electron microscopy to study the structures of Alzheimer tau filaments and Pick's disease tau filaments. After a sort of more general introduction, why believes that's relevant. In principle, you could do the same now for, you know, beta amyloid, for alpha-synuclein inclusion stuff. You can determine high-resolution structures of filaments from human brain without any intermediary steps, which I think is a step forward.
You just mentioned that this work doesn't immediately point to therapies, but has it pointed towards lines of research that might be followed more clearly to a treatment?
Personally, I think that in terms of, well, personally I believe that what one should aim for in the long run is prevent the disease. Because you could argue that by the time somebody has symptoms, even mild symptoms, there's probably already lots of these inclusions in the brain. And in order to prevent it, you obviously need something that you can give to people, but you also need to be able to identify people at risk, because only about 10 or less % are inherited.
Right.
And to identify people at risk, I think you need biomarkers, and probably something like PET ligands. And knowing the structures, for instance, in principle, should allow you to get better and more specific PET ligands. So that's one thing. The second thing, as far as treatments goes, is obviously the cryo-electron microscopy tells you what's there at the end. It doesn't tell you what was there at the beginning. But we believe that by knowing more about what's present in at the end for lots of different diseases, we may be able to design experiments to study what may have been there at the beginning, using, for instance, animal models or other experimental models. And so I think it's still early days, but I think in the long run this work will help a lot.
So you mentioned prevention and early diagnosis, I suppose. What is it known what types of things lead to these sporadic cases? I mean, what's the precipitating factors?
Well, I would have thought that what causes the disease in these people is the abnormal aggregation of these proteins. But what leads to the abnormal aggregation of the proteins
that's really what I meant.
Is another question. We just don't know. And it's also the question whether, well, I don't know, what the first, especially the first event, right? Whether you ever will know what the first, because it may just happen almost stochastically in a brain, and then given enough time, the pathology might spread through the brain. So understanding mechanisms of this so-called prion-like spreading, I think, is important, because that in principle would allow you to prevent the appearance of symptoms, because the very first events probably do not lead to symptoms. So people have small amounts of deposits in their brains, but they have no symptoms of the disease. But there could be multiple reasons that lead to these first events. We don't know.
You're talking about cell-to-cell spreading. It reminded me, I think John Collins did a meeting at Banbury on transmissible amyloid.
That's right, I was there.
You were there, that's right.
Yes.
So what's the, and I guess it got some coverage at the time, because the idea that Alzheimer's disease could be transmitted in some way.
Yeah, so that's the, I think it's true to say that when you have inclusions in the brain, the propagation of these inclusions through the brain is very similar to the propagation of prions. But that does not mean that Alzheimer's disease or Parkinson's disease are infectious. That's another argument, I think. I mean, we can talk about that, but that's another issue, I think. But I think the mechanisms involved in the spreading of the disease through the nervous system, they may be very similar. And I think it's a good sort of case for arguing that one should work on things that are biologically interesting, even when they're very rare. Because prion diseases are very rare. But, they may have thrown very important light on things that are very common.
Yes. Well, I think that's probably a good point to stop with, with a shout out for discovering fundamental research.
I think that's true.
Thanks very much.
Thank you very much. Thanks.